A white blood cell ate gas station yeast. Here's what happened to their organelles. MP is a 4-day-old macrophage, macro meaning large, phage meaning eater, a large eater cell, and is presenting to the emergency room with signs of an early cell death. Cell death in itself is not an unusual phenomenon. All cellular life has a natural time limit. Macrophages benefit from a type of programmed cell death called apoptosis. Apo meaning away, tosis meaning to fall. Apoptosis is a type of cell death program that describes a gentle shuffle off of this mortal coil. This type of cell death triggers when damage accumulates in the cell and prevents macrophages from attaining immunosenescence, a kind of sinister retirement plan. Immunosenescent macrophages become less able to carry on their duties, yet for some molecular reason are not prompted to die, causing a persistent level of low-grade inflammation. Apoptosis is a crucial mechanism by which the body prevents immunosenescence by removing and recycling cells that are past their best-by dates. Back in the emergency room, MP's membrane structure isn't entirely consistent with apoptosis. Upon death by apoptosis, cells will dissolve into apoptotic bodies, little bubbles of cytoplasm that prevent pro-inflammatory molecules from leaking out into the body. These little bubbles of cytoplasm are then cleared up by phagocytes like macrophages without any sort of drama. But MP's membranes aren't blebbing out into these little packets. MP's membranes look a lot more like Swiss cheese. But how did MP's condition degrade to this point? Looking back before admission, MP was a normal, healthy macrophage patrolling the skin mucosa right up until there was a breach in the skin barrier. MP was one of the many different types of immune cells that are called to action to protect the body from invading pathogens at the site of skin damage. Upon arriving on the scene, MP is met with a large variety of tempting targets. MP is a macrophage. Their job is to engulf microbes and digest them using powerful enzymes, acids, and superreactive superoxide chemical species. Upon recognition of these microbes' sugar-coated membranes, MP got down to business, polishing off yeast after yeast. After their meal, MP felt great. I have the best job in the whole body, they thought. Having their phagocytotic capacity reached, MP decided to take a nap and let their digestive enzymes finish the job. When MP awoke from their nap later, something was wrong. The hearty yeast meal MP consumed was still there. Not only that, but MP's body began to feel hot. MP's membranes began to swell as water flooded into the cell body. MP was then rushed to the emergency room where we are now. Laboratory analysis revealed that MP had failed to digest the fungi even hours later. Not only that, but MP's body was undergoing pyroptosis. Pyro meaning fire, tosis meaning falling from. MP was undergoing a programmed form of cell death, but not the nice controlled type of cell death apoptosis usually brings, but rather a type of cell death that is loud and terrifying. Pyroptosis occurs during infection and activates the protein complex called the inflammasome, which triggers the assembly and release of pro-inflammatory molecules whose job it is to warn the body of a serious problem. While pyroptosis can cause inflammation, which can be harmful, in this case, the danger is worth it. Forcing a timely immune response is key to ensuring that the infection is contained. What kind of infection is the body and MP fighting? MP ingested a fungus known as Candida albicans, a commensal microbe that is, in the majority of cases, not a present threat to the body. However, in regions of the skin that are moist and frequently experience friction, like the armpit, the delicate skin barrier can be damaged, allowing microbes to invade the body. I made a video on how the immune system determines whether a bacteria is good or not, and while I do recommend that you check that video out, the main point from that video that I am applying here is the fact that once any pathogen is on the wrong side of the body, it will be treated as an enemy, no matter the circumstance. Remember that the gut itself is, topologically speaking, on the outside. And while C. albicans is a commensal microbe usually kept in a harmless form by the slightly cooler temperatures of the skin and the chemical signals released from other commensal bacteria, if the conditions are right, it transforms into an opportunistic pathogen. A pathogen so potentially dangerous, macrophages will die attempting to warn the body of its oncoming danger. If macrophages are such ferocious eaters, why wasn't MP capable of digesting C. albicans? Macrophages make the protein dectin-1, a transmembrane protein that detects the yeast sugar beta-glucan and upon binding triggers a whole bunch of cellular programs that activate those strong enzymes and digestive processes. Candida albicans, however, developed a sinister trick. By hiding its surface beta-glucan with mannins, Dectin-1's ability to detect and activate digestive processes is then inhibited. This camouflage doesn't stop macrophages from eating the yeast, just from digesting them. Unfortunately for MP, once the cell signaling thresholds for pyroptosis have been reached, the process is nearly impossible to reverse. In just a matter of hours, MP's membranes will shred apart in a process called lysis, from the Greek term for loosening. And while it is unfortunate that MP will not make a recovery, we can rest assured that their demise was not in vain, as the inflammatory signals generated from pyroptosis will go on to protect the rest of the body. But our story is not done yet. An overgrowth of C. albicans rarely results in the infection of just one macrophage. MP is a different macrophage presenting to the emergency room. Only this time, MP's decline seems to be much more gradual. Pyroptosis still appears to be happening, but individual differences in macrophages and in pathogens can delay the onset of cell death to some degree. Imaging revealed something absolutely shocking was happening on the inside of this cell. The Candida albicans, which is usually in a single-celled form on the surface of the skin, was forming a multicellular colony called a hyphae within MP's cell body. Hyphae are fungal communities of individual yeast that form a pseudo-organism, sharing a contiguous set of membranes and even, to some degree, a bit of cellular specialization. This hyphal form increases fungal survivability by enabling these individual yeasts to collaborate, coordinating the production of enzymes and proteins that allow them to invade tissue and the production of biofilm to protect them from the body's chemical warfare. In this extraordinary case, however, the Candida albicans hyphae serves another key function: escape from the macrophage. As these filamentous colonies grow in size, they will push upon the macrophage membranes to the point of cellular rupture, killing the macrophage before the macrophage can kill it. Luckily for the human patient that housed both of these MPs, a surface skin Candida albicans infection is, for the most part, non-fatal in immunocompetent patients. While these MPs will not make a recovery, the patient will make a full recovery. In cases where Candida manages to invade into the deep tissue and get to the bloodstream, however, the story is very different. Candidemia, or Candida presence in blood, is a systemic infection that is difficult to treat and has a staggeringly high mortality rate. While most people have nothing to worry about here, immunocompromised patients who are uniquely vulnerable to fungal infection have to treat the possibility of fungal infection very seriously. Thanks so much for watching, take care of yourself, and be well.